This is currently an empty office. In a couple of months, it's going to be Cradle's new Mammalian lab. Cradle is an AI software company, but we have a lab here in Amsterdam. And now we're building a second lab, a Mammalian lab. And we're going to take you along through the entire process.
So at Cradle, we have built a software platform that enables scientists to design better proteins in fewer experiments with the help of generative AI. People are sometimes surprised that Cradle as a software company has a wet lab, but if you think about it, it makes a lot of sense. We're building models that enable scientists to develop better proteins using machine learning. But machine learning models have to learn, they have to train, they have to experience the real world. We want to make sure that every model that we make available is thoroughly tested in our own wet lab. You can think of it like a self-driving car company. You wouldn't sit inside a car if you knew that that company, the manufacturer, didn't actually have cars experiencing the real world. My name's Elisa. I'm a biologist by training and a co-founder at Cradle. Here I lead the customer success team, which makes sure that customers who work with Cradle end up meeting their goals and being empowered while they do so.
What we do in our lab is generating data that will help our ML team to decide whether new models that they develop are up to snuff. How that looks in practice, that when our ML team comes with a new model, they will have that model generate protein sequences. We build and test those sequences in our own lab. And then with that data, we can see, yes, this new model is doing better than the previous one. I'm Harman, co-founder at Cradle and leading the bioengineering team. When Elisa and myself had early conversations with the rest of the founder team, we discussed what our role will be, and we made, I guess, a very strong case that if we were to be successful as an ML SaaS company, that we needed our own lab to evaluate the models that we built. We are developing a lot of workflows and methods in our own lab. And because we're not generating IP in our lab, we can actually be pretty open about that kind of things that we do in our lab. So we publish our methods, protocols, workflows, so that our customers can benefit from them, take these workflows in their own lab and generate the data that our platform needs.
My name is Anja. I'm an antibody scientist here at Cradle, and my job is going to be to set up a lot of the functional assays here in the Mammalian lab. I think there are two main reasons why we want to have a Mammalian lab. The first is we need to test our proteins in functional assays to actually see if they have the same physiological activity that we want. Does the binding in SPR also correlate to binding on cells? Sometimes an epitope can be more available on a cell compared to actually recombinant protein. But one advantage of mammalian systems is that you have better folding of proteins, antibodies, post-translational modifications such as glycosylation, which contribute to better stability of your proteins. So generally, a lot of the antibodies you find in the field are produced in mammalian systems.
When I first heard that we were building a lab on the 5th floor of an office building, I was like, okay, yeah, we already have a lab on the 5th floor of an office building, but I do know already that it comes with a lot of challenges. And I was like, okay, yeah, cool, let's do it. On the other hand, I knew we would take on a lot. Yeah.
So now you know why we are building a Mammalian lab in our office space in Amsterdam. But building a Mammalian lab in an office space is much more complicated than you think it is. More on that in the next episode.